Okay, good evening. Welcome everyone to this ICTS Foundation Day. I'm glad you could join us for the celebrations, and I'm particularly delighted to welcome in our midst to join our ICTS family, of course, all the program visitors and others who are here for our workshops, but also especially Professor Jitu Mayor, about whom I'll say a few words soon. But welcome, Jitu, to the ICTS Foundation Day. And as you know, every year we celebrate this occasion of the setting up of ICTS. And we do it in the way which, in the spirit of ICTS, namely have some wonderful science lecture to stimulate us, and then afterwards a nice dinner. So food for thought, and then food after thought, and so on. so, so today, last year, we of course we were in lockdown, and so we didn't have this for a couple of years. And last year we had, of course, Ashok gave us the Foundation Day lecture, and so it's a privilege this year to have Professor Jitu Mayor. Jitu, I think all of you know, he is currently heads the NCBS, our sister institution. He's a cell biologist, and he is a chemist originally by training from IIT Bombay, and but obtained his PhD in the life sciences from Rockefeller University. And after a stint at Columbia and so on, came back to the NCBS. He studies, and we'll hear more about that, the fine structure, function, and dynamics of the membranes of living cells using methods derived from biology, chemistry, engineering, and physics. So a lot of the areas and a lot of things that interest people here at ICTS. So this has led him to explore how cells interpret information from their extracellular milieu, including interpreting the mechanical environment of cells in tissue context. His work has provided a new understanding of how membrane structure and composition are regulated in living cells, something very important, and I'm sure we'll get a deep insight into it after the lecture. And especially for his, he's been very well recognized for his work, which gives a deeper perspective on the functioning and roles of non-clathrin endocytic pathways, which I hope he will tell us what they are. I don't know. So Jitu is extremely well recognized for his pioneering work. He is an international member of the US National Academy of Science, a foreign fellow of the EMBO, a fellow of TWAS and the various science academies of India. And in addition to having won almost all the awards you can think of in India. And especially I want to mention as the Distinguished Alumnus Award from IIT Bombay as well. So the, but I mean, Jitu has been not only pioneering as a scientist, but also as an inspiration to the rest of us in the TIFR system for how he has led NCBS, for his leadership of an institution which is now one of the premier institutions in the biological sciences, not only in India but worldwide, and as a sort of, when I say sister institution, in some ways NCBS has functioned as a sort of a big sister to ICTS, and in the early days, and Spenta will be able to tell you more, but about how all the help we received from NCBS in having ICTS set up. But even for me, from my point of view, I should say, I've always relied on Jitu for sage advice and help, especially as we had to navigate various tricky issues. So it's been a pleasure to know Jitu, and I particularly want to mention one incident or one occasion. This was during these COVID times. As you know, it was an unprecedented situation for all of us. But Jitu very generously offered these COVID testing facilities of the NCBS to the ICTS community, which really helped us. I think that was an important step for us as we sort of tried to navigate and emerge from COVID and help us to sort of get our programs back on their feet. And this was very generously offered because they were doing it on a limited basis in their labs. And so to open up that that facility to the large numbers from ICTS was a very generous act. And so as I said, I really wanted to put these on record, and so now, without any further ado, let me ask Jitu to tell you more about his wonderful science. Thank
you. Well, firstly, Rajesh, thank you so much for that incredibly warm and generous introduction. Totally undeserved. But I was sitting next to the founder of ICTS, and I just noticed I wrote Founders Day. So Spenta, thank you for pointing that out. But I was particularly pleased to know that you're in the audience as well. So Rajesh invited me to give this talk, and all the kudos to me and all the blame to him. So I'm going to tell you about what I'm most excited about, which is thinking about the membrane of a living cell. The title of my talk, as you can see, is The Edge of a Cell. And I'm going to try and convince you that it is a living fabric. And it's something that, is an essential characteristic of a material that exists in a cell. And perhaps what we learn from the membrane will tell us about all sorts of materials that exist in living cells, as opposed to dead cells. Okay, so, and in fact, the subtext of the work is about how an engagement with physics has instructed us about understanding the structure and function of living cells, and this is not something new. I would like to start by telling you a bit about the history of our understanding of the cell membrane, some of the chemistry that I would like to explain in an evolutionary perspective, and then go on to tell you something about what we have discovered about the membrane by looking at it somewhat closely. So, with that little preamble, let me first of all acknowledge people who have been intimately part of this journey, especially in the recent times. So I call our group of colleagues the active actin-membrane composite team. We have theorists, and that's Madan, who needs no introduction at all. Madan and I have worked together on these projects on these ideas about the membrane for 20 years now. And in fact, I think very little of this work would have happened unless we had encountered each other. So Madan and his very merry band, Kripa, who's now a faculty at Azim Premji University, Amit, who's at IIT Delhi, Anirban, who's at Shastra University now. So these are people we worked with closely in some of the work that I'm going to tell you about from our work. The chemists at the IIIM in Jammu, Ram Vishwakarma, Parvinder Pal Singh, and his colleagues who are quite numerous, so I haven't put their pictures up here, but there's a whole group of chemists who have helped us in our work. Kabir, who has been a bridge between the theorists and the biologists for many years when he was a student between Madan and I. And we have a whole range of biologists who have worked on this, beginning with Darius and Abrar, Ria, Ambika, Suvrajit, Joseph, Chandrima, Saryu, and Thomas. And I'm going to mention their names as we go along, because the work that we've done is, has been an exciting journey, possible only because of this an amazing group of people I've had the privilege to work with. So that's what we've been looking at. The membrane of a eukaryotic cell. It is a multi-component fluid lipid bilayer. It in fact, at closer range is in cartoon form is expected to look like this, it has a patchwork sort of quilt description or a distribution of molecules. I mean, this is a cartoon, but it's a reasonable cartoon of what the membrane looks like. And the question is, how does this sort of cartoon emerge? But also, I should point out that what this cartoon tells us is that there are a number of different components in the membrane. In fact, the membrane of a living cell contains over 1,000 different individual species of molecules. It contains sort of 5 classes of lipids, and I'm going to tell you something about them in a while. But it's an incredibly complex chemical mixture. However, it is exquisitely organized. And that's sort of, in some way, I've given the story away, but that's what we're going to talk about. Okay, so the first encounter that the membrane of a cell had with I would say, physicists was when Gorter and Grenville used sort of tools and methods developed by Agnes Pockels of the Pockels cell, and Langmuir, a chemist looking at the physical properties, physico-chemical properties of various hydrophobic substances. The Langmuir trough is one of his inventions. And they discovered that if you took a red blood cell and extracted it with lipophilic solvents, you could extract lipids and if you put lipids from that red blood cell on a Pockels cell or a Langmuir trough, you could measure the area occupied by these molecules, and if you knew the approximate molecular weight of these lipids, then you could determine the area of the cell. And it turned out that the area occupied by and if you image the red blood cell, which you could at that time, you could calculate the area of the red blood cell, but the lipid lipid area was twice that of the red blood cell. So they proposed that the lipids must be organized as a bilayer. In fact, the bilayer was proposed in 1925, almost 100 years ago. And then the fact that the bilayer had proteins associated with it was, again, hotly debated. People said there were proteins, separated from the bilayer. Then this trilamellar form of a bilayer was proposed. And then about 50 years ago, Singer and Nicholson proposed what is perhaps what the model of the lipid bilayer that most of us study in textbooks today. And that's the fluid-mosaic model, which synthesized observations of membrane protein distributions from both electron microscopy analyses as well as analyses of protein diffusion in the membrane, and synthesizing those with an understanding that the bilayer was a highly complex mixture. They suggested that you had proteins dissolved in a sea of lipids or in a solution of lipids, which indeed were organized as a fluid bilayer. And in fact, that's their description here, which at higher resolution looks a bit like this. You have proteins sort of embedded in a bilayer in the matrix in which you have a sea of lipids. And this was in a well-mixed system. Proteins did all the business end was the proteins, and the lipids were just this background solvent. And of course, I'm going to convince you that that's not the case. But what are these lipids? So there are phospholipids, there are glycolipids or lipids that contain a phosphate attached to diacylglycerol, or phospholipids attached to ceramide backbones, and glycolipids that are also attached to ceramides. I mean, these are names and they can remain so, but just to appreciate that there are 5 main classes of lipids in a eukaryotic membrane, and we'll address this term eukaryote in a bit. And there's also this enigmatic molecule called cholesterol. So that's sort of effectively, if you reduce and look at it from coarse-grained chemistry, that's sort of what you can do to a membrane of a living cell. You have 5 classes of lipids and a sterol molecule mixed together, forming this bilayer. Now, I think as you know, one should ask the question: where do these lipids come from? And of course, nothing in biology makes sense except in the light of evolution. So we're going to explore the evolutionary time over which these lipids arrived in the eukaryotic cell membrane. So here is sort of what an evolutionary tree of life looked like about 10 years ago, where there was an original ancestral cell, and that original ancestor was the ancestor of all life forms. Prokaryotic life and eukaryotic life are the 2 types of life forms that we know of. And of course, in the prokaryotic life forms, there are the bacteria and there are archaea. And the eukaryote is supposed to have been derived from a synthesis of some bacteria meeting, some bacteria being engulfed by another form, and the other form was supposed to have been an archaeal form. But the eukaryote was placed on a limb here, suggesting that the eukaryote was derived from an archaeal form. It shared the protein synthesis machinery, it shared the DNA replication machinery with archaea, and in fact, the bacteria was the stuff that contributed to the mitochondrial apparatus of a eukaryotic cell. But lipids, as it turned out, were a very different, looked like they'd come from a very different space. The archaeal lipids had a chirality which was the opposite of what the eukaryotic lipids carried. They had enzymes that were actually not present in eukaryotes. In fact, the eukaryotes had enzymes that were more similar to the bacterial lipids. And this didn't make sense, because if you had the protein synthesis machinery, you had the DNA replication machinery, and you had everything else from the archaea, why were the lipids coming from the bacteria? And in fact, that remains an unsolved question until very recently. In 2015, in a deep vent somewhere in the Arctic Sea, a huge efflorescence of these archaea were discovered, and they were called the Asgard lineage. And it turns out that one can identify very closely, somewhere along this lineage, a archaeal origin of the eukaryotic life forms, which then supposedly fuses with a subset of bacteria called the alpha proteobacteria, resulting in what you have are all of us here today. And therefore the lipids could be contributed from any of these parents, and of course, as it turns out, the eukaryota carry lipids from the bacterial lineage, but they also carry one more compound, which is sterols. Now, sterols are also present in bacteria. They're completely absent in archaea. And the sterols have a funny story, which I want to relate in a bit, because the sterols are not lipids. They are these polycyclic molecules with a hydroxyl group at one end, capable of hydrogen bonding with many of these lipids, and they completely alter the physical structure of the membrane. They make the membrane more fluid, and I'll explain in a little bit what else they do. But it turns out that the cholesterol and sterols, molecules of that kind, could never have emerged in early life forms when bacteria emerged. Bacteria emerged in a time on this planet about 3 billion years ago, or 3 or 4 billion years ago almost, when the partial pressure of oxygen was about, more than 10 to the minus 10th of what it is today, or less than 10 to the minus 10 of what it is today. And due to O2 emissions, if you please, not CO2 emissions, O2 emissions, there was dramatic climate change over the billion years, over several billion years, leading to a point where there was sufficient molecular oxygen available for the synthesis of complex lipids like cholesterol. So the fact that there was this kind of climate change produced the invention of, or manifested in the invention of sterols, which emerges actually after oxygenate photosynthesis and the oxygenation of atmosphere and oceans around 2.7 billion years ago. So, and the reason for that is because many of the synthetic steps that are involved in building these sterols require, not many. I mean, the steps involved in the synthesis of cholesterol require about 11 molecules of oxygen. Again, I'm not going to belabor this point, but just to say that this was a fantastic co-evolution example of chemistry and its cooperativity with lipids, where sterols, the argument being that selection pressures rendered the sterol molecule more and more competent for membrane function, which required a huge diversity of possibilities. I should mention here that the bacterial membrane is sandwiched with its cell wall, and therefore the bacterial membrane, in principle, doesn't do very much. It doesn't undergo all this it doesn't produce all these fantastic structures, ruffles, and things like that eukaryotic cells do, because the bacterial membrane is actually pretty well stuck to its cell wall, and therefore it doesn't use the capacities that the chemical sterol provides for the particular physico-chemical structure of the membrane. Okay, so now moving on with this revised evolutionary tree of life and our understanding of the lipids, of where these lipids came from, I've sort of paid obeisance to Dobzhansky, and now I can tell you what in fact we think the membrane looks and feels like. Okay, so at this point, again, when the structure of lipids and the composition of the bilayer was known, the membrane was again, because of a physico-chemical expectation, thought to generate different phases. So phase segregation of these multi-component systems began to be examined in great detail when it was found that if you took sterols and put them into lipid bilayers, which had multiple components, you could now prevent these components from phase segregating into solid gel-like phases or simply liquid crystal disordered phases. But instead, one could produce a distinct domain called an ordered phase, or a liquid ordered phase of these membranes. So the sterols actually provided the membrane to build what are ordered domains and disordered domains. So bypassing, if you will, the gel phase. If you in fact produce a gel phase, you'll in the presence of these disordered domains, the membrane will no longer maintain its permeability barrier. So in fact, if you produce gel phase in membranes, the membrane becomes exquisitely permeable, and therefore, all life will stop. Especially if you produce it at the cell surface. So in fact, cells try and maintain a lipid composition where the gel phase is prevented from taking place. And sterols also provide that capacity to buffer the presence of these gel phases and make these liquid ordered phases, which make sort of for a liquid order-disorder coexistence. Now, this was all work in vitro, where people were looking at artificial membranes. And of course, that didn't escape the biologist who then ran with this idea that membranes in living cells must also produce phase-segregated domains. And these phase-segregated domains would have functional properties, which would then carry out all kinds of fantastic things. So if you had a membrane of particular composition, if it phase-segregated, now in those phase-segregated regions, you could accumulate, say, for example, if membranes phase-segregated in the Golgi apparatus of the cell, those membranes could be directed one direction to the cell surface, whereas if the remainder would go to another part of the cell. So you had a way of segregating and sorting molecules, or you had a way of concentrating proteins into specific regions where these ordered domains could accumulate molecules of particular type. And so these domains were called rafts, and the rafts sort of became the sort of poster boy of cell biology, or I shouldn't say poster boy, poster person of cell biology, which for a very long time, and almost everything that we know about sorting and signaling were thought to be explained in terms of these rafts. Now, what I'd like to say is that perhaps these phase segregation, and phase segregation is also re-emerging in the form of biomolecular condensates, with, I think, very much the same sociology. I mean, the biomolecular condensates of today, in my mind, resemble the arguments and concerns we were having 20 years ago when rafts were being put forward as a central paradigm in understanding cell biology. And I think that's instructive for us to think about some of the sociology of these encounters between physics and biology. Because I think simplified ideas or simple ideas in physics don't naturally translate into a biological system. And I think that was one of the things that I took away from this whole encounter with rafts. But let me say that in the cell we do know, and this is from biochemical measurements, that the composition of membranes in different parts of the cell is exquisitely and tightly controlled. And of course, these compositions, because they are tightly controlled, they will exhibit unique characteristics and unique properties. And of course, they will probably exhibit different phases in cell membranes. But as I just mentioned, that to produce phase-segregated regions in cell membranes, one would need a particular composition, and also if processes are occurring at thermal equilibrium, you'd also need a particular temperature. So composition and temperature are intertwined, and I don't need to tell this audience that, but phase segregation will occur as a consequence of temperature and composition. But as we know, most cells, whether it's in our body or elsewhere, exist at a whole range of temperatures, although we as eukaryotes or multicellular eukaryotes are temperature buffered. We also have very large ranges of temperature that our cells exhibit. Yet the notion that phase segregation being driven by thermodynamic principles would operate in membranes became a dominant paradigm in understanding how cells function. And I want to sort of say that argument doesn't hold much water or much lipid because of some experiments that we did when we started looking for these membrane rafts in cell membranes. In living cell membranes. So about 20 years ago, we wanted to ask whether we could understand the picture of the membrane that was presented so nicely by a colleague of mine, Michael Edidin, where lipids and proteins were present in these beautifully segregated patches. And one of the components that was part of this segregation was supposed to be segregated were proteins called GPI-anchored proteins. These proteins are proteins that are attached to glycolipids, which sit in the outer leaflet of the cell membrane. And they are supposed to form segregated patches because they are trafficked in a very unique way to one domain of a cell and not the other. So we thought looking at the distribution and organization of these GPI-anchored proteins would be a very nice way to explore the structure and the organization of phase-segregated domains in cell membranes. So, so we put put these molecules to work by putting some binoculars on them so that they could look for their neighbors. So we set up experiments, and we built some tools to study the organization of these molecules. We're using Foster's energy transfer imaging technology or methods, where we could ask the question whether these proteins in the membrane of a living cell could sense their neighbors or not. And they could sense their neighbors. If they sensed their neighbors, they would exhibit certain characteristics of fluorescence, which we could then monitor. So in fact, I won't be so enigmatic about these methods, but it's, I mean, just in the interest of time, I'll just say that what we established were methods to measure the close interaction of these proteins in a living cell membrane in real time by first exciting these proteins with polarized light. And when these proteins give out a polarized emission, if they encounter a neighbor within 5 nanometers of the molecule, and that's the scale of the molecule itself, 3 to 5 nanometers, the fluorescence from the excitation of these molecules becomes highly depolarized. So by simply monitoring the depolarization of the fluorescence emission from these fluorophores in the membrane, we could tell whether you had fluorescent GPI-anchored proteins near to their neighbor, have near neighbors or not. Or if they were very far apart, you'd see very high polarization. If they were very close by, you'd see low polarization. So using that as a metric, we started exploring the details of the structure of these molecules in living cell membranes. And if you now focus on that green little square there, the fluorescence intensity image looks completely flat. But if you look at the FRET image or this polarization image, what you see are incredibly rich structures. These structures correspond to these GPI-anchored proteins organized as incredibly tiny clusters in these little blue patches where there is extreme fluorescence depolarization. And in these patches right next door, the molecules are almost monodisperse. They don't form any clusters. These clusters exhibited, were not only discovered by us, but we described them very early on. They were described by many others by using many other techniques, and they remained quite a mystery for a while. They exhibited extremely peculiar properties. They were, first of all, spatially heterogeneous, as I showed you here. There was concentration independence, meaning the clustering that these molecules described were relatively independent of the concentration of the protein in the membrane. Of course, if you went to very high concentrations, you would see trivial effects of crowding. But the clusters were formed, and they were actually forming and breaking up quite happily in very rapid time. They formed and broke up, in fact, in these particular patches with a frequency of around a second or so of these little clusters. What was more important is that this clustering was maintained actively. It wasn't present in a membrane in a dead cell. These clusters were not present when the membrane of the cell was disconnected from the cortex that the membrane is draped over. So if you made these little blebs, which is the membrane of the cell, and the blue, so the magenta color here is in fact the cortical actin cytoskeleton, the armature over which this membrane is draped. Those blebs that are produced, in fact, lack these clusters. And we then began asking ourselves, what happens when the membrane encounters the actin cortex? And here, we some really excellent work from Aki Kusumi, who's a colleague, in fact, now in Okinawa, and he showed that when the membrane is spread out over the actin cortex, it senses the actin cortex as a frictional barrier for diffusion of molecules in the membrane. In fact, and that's a passive sort of engagement with this frictional barrier that the membrane senses when it is draped on top of this meshwork of actin. And he showed, using some very nice experiments, that the presence of the membrane on a bilayer generated little compartments through which molecules can hop and move. And when the compartments break open, they actually start exploring wider territories. Now, but that was unable to explain what we were interested in. And then there was a third encounter. And Madan and his colleague Kripa said, well, let's start from first principles. What you have here is a membrane that's sitting, that is being templated by dynamic cortical actin. And if the cortical actin is in the form of small filaments, and it is connected to actin, it's connected to motors, or myosin motors, the membrane is going to exhibit, the actin filaments are going to exhibit clusters such as this. So he and Kripa, he proposed I think a very simple framework for us to understand this. And I think this physical framework that Madan and Kripa proposed has led to a number of experiments that we have taken cues from this theoretical framework, which have allowed us to understand what's happening in a living system, a living membrane. And I think I'll go through the proposal here. What he proposed is that there are two key elements. One, there are two types of actin filaments, or two types of filaments, short dynamic filaments and a static mesh in which these filaments are embedded. And then there are motors that allow the filaments to slide against each other and be advected as well. And if that happens, if that is the case, then one can write down both equations for the filament orientation, which are connected to how the filaments slide against each other, and the contractility of myosin and the orientation of the filaments are governed by these sets of equations. And then filament concentration can be is exquisitely dependent on the active advection of these filaments by the cortex, as well as the diffusion of these filaments in the subcortical region. So when you write these down, numerical simulations gave us the picture that we were looking for. So the numerical simulations of these actin filaments, if they were coupled to structures in the membrane, they produced these kind of patterns that we, and in the presence of what Madan called active noise, that is the natural spread in the distribution of the cortex and in the cortical activity of the myosin, you created these sorts of patterns where things were moving about and jiggling about in space and time. And they created the patterns that we were seeing in the cell membrane itself. The model actually exquisitely recapitulated the experiments that we had done and the measurements that we made. I will just show you pretty pictures to say that that's what they look like. In fact, there is very little difference between the experiment and the theory in terms of what the theory could explain in qualitative terms. The model also made very key predictions. It predicted one, of course, trivially, that if there is a direct coupling to the cortical actin, that should also drive clustering. I mean, remember, we are looking at molecules sitting at the outer leaflet of the cell. But if you had a molecule like this in the membrane, which is now coupled to the actin cytoskeleton beneath, these molecules should also form the clusters that are exhibited by the molecules that we are looking at. And of course, that's exactly what happened. These, any molecule or any protein in the membrane that is connected to the actin or that can be connected to the actin, and in the case that we were trying to prove it, we synthesized these molecules, made model proteins, and generated these clusters in the membrane of these proteins, again, using the same tools that we had developed. It also predicted that there would be a signature in the fluctuations of the concentrations of these molecules, which would be consistent with there being an active driving of these molecules in the underside of the membrane. And that signature, well, of course, if a system is well mixed, you'd expect these molecules to, the fluctuation in the distribution or the density of these molecules would go as a square root of n. But with activity, one would expect something different. And so we set up experiments to measure these fluctuations in the membrane, and therefore when we found that without activity, you extracted the root of n dependence on the density of the molecules in the membrane. But with the activity, now the fluctuations approach that of what the theory predicted, which was to the power of n to the power of 0.8. So the predictions of this model were borne out by some of the tests that we did. And in fact, that led us to now ask questions which were more ambitious and said, if this model is that simple, can we now reconstruct this whole system in an in vitro system? And in fact, what we ended up doing was building a in vitro setup where we could then look at the actin cytoskeleton, the myosin motor, and lipids in the supported lipid bilayer, and generate exactly the kind of clusters we were seeing in the membrane, which had all the quantitative characteristics we expected from this active driving of clustering in a membrane at the underside of the membrane. Of course, the scales at which we were looking at this were much larger than that of the cell, because the components that we were using to build actin and myosin were also much larger. We were using actin and myosin filaments that were maybe 10 times the size of what was present in the cell. But the principles were still quite evident. Now, so then the other question we asked is, how do these GPI-anchored proteins connect across the bilayer? For transmembrane proteins, of course, you have an actin binding domain here, but the GPI-anchored proteins are sitting at the outer leaflet, as I mentioned when I told you about these explorations that we had started. And so to understand that, we again turned to some chemistry from Ram Vishwakarma, and we started, we made synthetic analogs of these GPI-anchored proteins. When I say we, I mean Ram and his colleagues, made synthetic analogs of GPI-anchored proteins. And then Ambika and Ria in the lab tested out these synthetic analogs and found that you needed long saturated acyl chains at the outer leaflet to couple to the inner leaflet. And that required a lipid at the inner leaflet called phosphatidylserine, or PS, which could also connect to the lipid at the outer leaflet to the lipid in the inner leaflet. And if you had the lipids in the outer and the inner leaflet, which had long saturated acyl chains, cholesterol, we're coming back to cholesterol. Cholesterol was necessary for this coupling to take place to build this bridge between the outer and the inner leaflets, outer and the inner lipid species. So cholesterol is required to construct this. And of course, we did some simulations of a membrane bilayer. And at the temperature at which we were making measurements, the membrane bilayers themselves are well mixed. because at this temperature at which the cell exists, the lipids and their composition are in fact going to exhibit well-mixed property. But if you now hold some of these lipids, which have long saturated acyl chains at the inner side, with the actomyosin machinery, now you end up recruiting the lipids that have long acyl chains in the outer leaflet, and they connect to the lipids that have long acyl chains in the inner leaflet, forming these little patches in the bilayer, which are little assemblies of ordered domains in the membrane. And these patches now are expected to exhibit an ordered patch. And they are now they're producing an ordered patch in a membrane at temperatures at which these ordered patches should not exist. Right? So they're overriding these any thermodynamic consideration of this so-called phase segregation that should result in some of these ordered domains. But these are being generated by a different machinery. They're being generated by a mechanism that the cell is building. So now, of course, we looked using a solvatochromic probes to ask whether we could now detect these ordered patches of membranes in the cell and whether they coincided with these clusters of GPI-anchored proteins. I'm going to speed up a little bit because we're already at 50 minutes. and I've got a few more slides to go through. Yeah. So what we found was that the GPI-anchored proteins at the outer leaflet produce these clusters. And now if you correlated these clusters with the order of the membrane in those regions where the clusters were present, we found that the clusters were present where the membrane was more ordered. Right? So this has given rise to a new way of looking at how this active machinery generates these ordered patches in the membrane. So if you have a membrane which is well mixed, if it couples to the actin cytoskeleton beneath, it produces these little nano-clusters driven by the actin itself. And those nano-clusters now, of course, they will exhibit enhanced or lateral interactions with other components in the membrane, therefore generating what we would call regions that are like active emulsions. And these active emulsions, I would contest, are the stuff of all things that are going to regulate the heterogeneity that one sees in the bilayer. So these active emulsions now are emerging, if you will, as a way of understanding how this organization of the membrane is being developed by the engagement of this entire subcellular machinery. In fact, it's the composition in conjunction with this, with the driving forces of the active mechanics of actin that creates these patches in the membrane. And I think there's fair degree of both theoretical and experimental evidence for this now. So membrane, as I said, membrane heterogeneity is now coming up as a consequence of the active mechanics of cortical actin. And any membrane protein which has an actin binding domain will be subject to these forces. As I mentioned, lipids that are the special class also are exhibiting such driving, and so do glycolipids. Now, so we now come to what we think is the 21st century model of the membrane, where you have three classes of membrane molecules with respect to the dynamic actin. The first is what I think Singer and Nicholson were actually looking at, because they did measurements primarily on extracted membranes or membranes that were not necessarily living. In the living membrane, you get these specific environments which are generated by these driving machineries. And of course, if you have driving machineries, they will naturally, and they are subject to regulation, because you need to make the machinery, you can switch the machinery on, you can switch it off. So there are a third class of components, which we call the active components, which are regulators. And I think the next phase of what we would like to understand and in fact, I'm going to give you some evidence for that, are the roles of these active regulators. I think this is the most exciting thing, that the cell is now able to regulate how these systems are produced in the membrane at will. I'm going to actually give you an example of a particular regulator, the integrin receptor. This receptor senses both the chemical and the physical environment of a cell. It senses what kind of substrate, the chemistry of the substrate, and it also senses the stiffness and if you will, the corrugation or aspects, mechanical aspects of the substrate, and the information that it gathers, it actually acts to influence the membrane. The receptor itself is a complicated, heteromeric molecule, binds to specific ligands on the outside, which are called variously extracellular matrix ligands, and it activates a whole signaling cascade beneath. This receptor senses both ligand type, receptor type, and it senses a wide range of chemical species on the outside. But it also exquisitely senses the force that the cells need to exert in order to move somewhere or in order to remain stuck to a surface. And what we discovered is that when you put cells down on a surface that carries a ligand for these receptors, a stiff surface that carries ligand for these receptors, you end up generating a huge number of these clusters in the membrane. So the cell comes down initially without too many clusters, and then when it lands on the surface which carries the ligand, it generates a fantastic amount of these clusters. So these big blue patches are effectively that. So the membrane gets filled with these clusters. So using such a description, we actually found that not only were specific ligands necessary, in fact, the cells require a specific molecule on the outside called fibronectin, but not labyrinthine collagen. Therefore, the cell can discriminate between what it's going to do to the membrane using these receptors. But it also utilizes a mechanotransducer, which is downstream of the receptor. The mechanotransducer sits somewhere here downstream of the receptor to generate these patches in the membrane. So, of course, using such an assay, we were able to look at the parts list that is required for the generation of these clusters. I won't go through the details of this, but just to say that that this whole system has actin remodeling agents. It has connectors that connect molecules from the outside to the actomyosin machinery on the inside, and it has the regulatory machinery that's responsible for doing all these things. And we can take this parts list and ask the evolutionary landscape and say, where do these parts lists exist in the evolutionary tree of life? And as it turns out, all multicellular creatures have an abundance of these part lists. And many single-cell protists have smatterings of this, but the data from the single-cell protists is not yet sufficiently rich for us to mine it and give concrete statements about whether these part lists exist there or not, but I suspect they do, because we do find unique features that are present everywhere. So I'd have to say that this active composite membrane has been active for a very long time, at least 1 billion years. Finally, let me sort of end by saying why should this matter for the cell? Right? Why should this active composite system matter at all? Why not just use a membrane which is passive and just supports the protein fabric that is part of the signaling system? Well, clues for that actually came from the same experiment that I described to you here, when we discovered that when the cell began spreading explosively, it actually shifted from being from not having any of these clusters to having a large number of these clusters. And using genetic methods, we could actually switch off the production of these clusters by changing the chain lengths of these lipids inside the cell. And when we did that, we found that the cells no longer exhibited any of the spreading response that we found. And this, to us, suggested, sort of in a diagrammatic form, I'll explain this, that when the signaling activity takes place, it produces this liquid ordered patch in the membrane, which in some sense encapsulates the information from the chemistry and the mechanics of the mechanical environment that the cell is sensing, and generates a sort of, in some ways, a memory of that environment, which then recruits effectors which allow the cell to do something. In this case, spreading. In other cases that we discover, it allows the cell to walk up a stiffness gradient or migrate in a particular direction by encapsulating that information from the outside into some configuration of lipids in the membrane itself. And in fact, so this cycle of sensing the substrate and changing the properties of the membrane in order to respond to it is a consequence of this active composite. Right? There's another consequence of this active composite and this composite here is, I've just given it one dimension, which is the ability to produce these liquid ordered domains. And these liquid ordered domains have another possibility, and then that is when they create these ordered domains, because they're created in a kinetically determined fashion, they will drive conformational changes of molecules that sit in those patches. And those conformational changes of molecules, again, have consequences for the structure and function of proteins in the membrane. And this is an area that we are incredibly excited in exploring and hope we will be doing this in the near future. Here, in fact, so we've developed some new tools which allow us to image these domains in real membranes by building chemical probes, again with our colleagues in Jammu, and Chandrama in the lab has been able to show that you're now watching these liquid ordered patches in real time. So this is a movie of those liquid ordered patches as illuminated by the solvatochromic probes, and where the red patches now reflect these ordered domains in the membrane, and the blue and green patches are the ones which are more disordered. And so now what we'd like to do is, of course, couple the organization of proteins, their conformations with this sort of imaging approach and see what happens. And this is important because many receptors in the cell membrane that sense the outside world for the cell and make it do all the tasks that a cell does, are embedded in this bilayer, and they all activate machineries that are capable of generating these lipidic domains and lipidic patches. So I think the active composite is going to be very important in the regulation of their function as well. So let me end by saying that, I hope I've provided you a reasonable exploration of the evolutionary history of membranes and the components that give rise to the membrane and its structure. And finally, what the membrane is doing in conjunction with this one tiny part of the cell, which is generating these little patches in the membrane. But the membrane, as I said, sits on a very dynamic actin meshwork, and that actin meshwork is modulating the shape and form of the cell in real time when it itself is modulated. And that's a subject of stories many stories for many other days. With that, I'm going to say that I hope I have been able to convince you that compositional control, the regulatable assemblies, can give rise to active chemistry and information processing in ways that we have so far not considered by by the utilization of some of these methods, mechanisms that the cell has deployed for the control of these structures. So in conclusion, I'll also say something about when physics meets biology at the cell surface. Of course, there have been multiple encounters. And they do reflect an intertwined relationship between concepts borrowed from physico-chemical studies on artificial membranes, fundamental insights from theory, and reality checks with experiments on membranes of living cells. And of course, all this in the light of the evolution of the system. So the edge of a cell is a living, breathing skin, a mechano-chemically regulated actin-membrane composite, where I think we have principles of orchestration, self-organization, and these are going to have, and we need to explore this now, consequences on many of the functions that the cell exhibits: migration, signaling, fate, and of course, building tissues. So with that, I'm going to stop here by saying it's been an extraordinary journey with some extraordinary people. These are the sets of people I've talked about, many of them, and their work, at least showed pictures of them, while they have gone about doing what they like doing. This work would not have been possible without, of course, having this fantastic group of people who support us at NCBS from our facilities. The work that I described on the evolution of the parts list was in fact a rotation project with a few students at KITP, Quantitative Biology, in 2015. And we hope to take that work further in my own work. And much of the work that I described about building the artificial system to explore membrane organization was actually done when I spent several summers at the Woods Hole lab near the MBL labs at Woods Hole in Massachusetts. So thank you very much for your attention. I'm sorry I got a bit carried away, but...
No, not at all. Thanks a lot, Jitu, for taking us through this long journey, and especially with the interaction with physics, both the cautionary aspects as well as finally the successful aspects of the collaboration. And I hope there will be more of that, and perhaps some of you, the physicists in the audience, might be inspired to sort of take look into questions in biology. So before we ask Jitu, I'm sure we'll be happy to take questions. But before that, I'd like to invite the founder, since it's the Founders Day, to present a memento on behalf of ICTS to Jitu.
Okay. Must be a spring, I'm sure.
Just a little.
Oh,
Sandalwood.
wow. Thank you. Beautiful. Look at that. Thank you so much.
Our pleasure.
May there be peace, is what
Yes.
the Buddha would say.
Yes. So we can have some questions from the audience. So yeah, Vijay, then Rama.
Hi, Vijay.
Hi, two questions.
Yeah.
So one is about the evolutionary aspect. So you said when you were talking about the composition of the membrane that something was inherited from the archaea and something from... What about the cortex? Was that something independently developed, or are there signs that was also borrowed?
Fascinating. The cortex actually is present, the cortex and its cortical machinery, the actin, myosin motors, are present in sort of not in the same kind of structure that they are in eukaryotic cells, but in bacteria, for example, you have examples of filaments like that look like actin. In archaea, you're beginning to have examples of things that could behave like motors, right? And so the cortex is emerging actually from a combination of these two life forms, it's very exciting work right now that's happening, that's looking at these questions.
Second question. When you said the actin composite membrane,
Yes.
there are membranes which don't have an apparent actin structure to Golgi,
There are membranes, yes.
So, but possibly you meant something which is driven by non-equilibrium processes rather than actin.
Yes.
Per se,
So the plasma membrane or the outer membrane, I'm arguing, is this active composite. But other membranes in the cell, we think are operating operating on very similar principles because they engage with many cytoplasmic elements that impart similar kind of activity to the constituents. But I think not just actin, not just membrane, DNA as well, right? I mean, DNA is a one-dimensional polymer. It also engages with motors that are driving it, and I think similar principles are emerging even there. So I think any sort of fabric, any sort of material that's being built by the cell will have properties such as this.
Thanks.
Rama.
Yeah, so when they say that too much cholesterol is bad for us, do they mean it's because there's going to be too much clustering of the GPI proteins or too much ordering in the lipid layer?
But first of all, who's they?
Hey, the doctors.
Okay. No. I can't say what cholesterol is going to be doing. If you take away cholesterol, all this coupling between the outer and the inner leaflet disappears. If you add too much cholesterol, you end up, and of course, at the same time, increase the number of lipids that have saturated tails, long saturated tails. The membrane ends up being very easily sent into this ordered phase, and that can be extremely detrimental to many signaling reactions. So, I mean, I can't comment on what happens to us when we eat too much cholesterol, but when a cell modulates, cell seems to keep its membrane cholesterol very tightly regulated. In fact, that's the other very interesting things about these lipids. They are maintained with an exquisite sort of compositional, sort of compositional control. And how that is maintained, we have no clue. I mean, at least in the eukaryotic membrane. In bacterial membranes, we have some idea that there are sensors of certain physical properties which then feed back into synthetic pathways and maintain their structure and composition. But the environment sort of impinges on these membranes by the food you eat, right? And that changes the complexion or the composition of the membrane too.
Riddhi.
Thank you for such an informative lecture. I'm not a biologist or physicist or chemist. But so you talked about any living cell. This is the composition, or you study specific
so
cells?
So what I've talked about, I mean, of course, whenever you look at anything in biology, you have to look at something specific. So we've looked at a range of cells. Most of them are cells from vertebrates, and some from fruit flies. So we've at least looked at these two types of cells. Many of the cells that we've looked at are actually live as single cells. They don't live as in tissue form. So those are the sets of cells we've looked at. I mean, the names would be almost irrelevant, but these are the types of cells that we've looked at.
So if you look at, say, cancerous cells,
Yes.
what is the changes that
There are
would
dramatic
occur?
changes in these
Cell membrane.
properties when we look at cells that have sort of generated certain types of tumors and certain types of systems. But there is no systematics yet to when we've made these comparisons between different types of cancers and different kinds of perturbations, we have not yet established any systematics that we can make sense of yet. But there are changes.
So are there any indicators as in the cell membrane which would indicate that it could turn cancerous, or there are some markers?
Well, that was one of the the paths that we trod, but we couldn't find anything systematic at this stage, because perhaps we don't know what we are looking for at the moment. On the other hand, when we've looked at cells we've looked at specific changes in the membrane, for example, in cells that have acquired certain kind of lipid compositions that are aberrant. When cells have acquired an aberrant lipid composition, their capacity to signal from the outside changes dramatically. So, for example, under certain aberrant lipid composition conditions, the insulin receptor becomes completely silent. So the cells acquire insulin resistance. And now if you change the composition, the insulin receptor wakes up again.
Oh.
And that seems to be due to some of these mechanisms.
Thank you.
Yeah, I had a related question actually. When cells die, is there and you briefly mentioned, I think, in future directions also, but is it that they die from the outside or from the inside, so to say?
So how do cells die? No.
I mean, from the cell membrane starts becoming less functional.
Yeah, but I think that's a consequence of certain. So there's this programmed cell death, right? Which actually, as part of the program, one of the mechanisms is to dissolve the cytoskeleton, and the cytoskeleton starts falling apart. The membrane starts producing blebs, and the asymmetry of lipid composition that is there in the membrane is completely destroyed. So many things happen but the cell seems to have a program that starts to work to shut its insides down.
It starts from the inside.
Yeah. And there are many programs, but these seem to be, of course, triggered by external, for example, energy deprivation, hypoxia, all sorts of things. Environmental
changes.
Yeah. So the structure that we see in
where?
Yeah.
So the structure that we see in the cell wall is also dependent on the ambient flow. For example, there is a lot of salts and water, and the flow of them, the structure depends on them, whether the flow is turbulent or laminar kind of thing.
Right. So the cells do respond to the physical flows on the outside. They do respond because they have sensors, if you will, of flow, which can then change signals that are transmitted to the membrane, and the membrane also responds accordingly. So there are physical cues from flows, whether it's turbulent or laminar. I don't. They will have big differences in what the cell senses. But when you look at flows, you're looking at shear flow, shear on the surface of the membrane, and that definitely has an impact on what happens to the membrane.
So in our body, different parts has a different kind of cell.
Right.
So the structure or the size of the cell is dependent on the flow.
The size, I don't know, but the cells, cell size is regulated by other mechanisms.
Not
necessarily flow, no.
But it is dependent on the cell wall, how cell wall is.
It certainly senses the flow. For example, in a capillary, you have endothelial cells which sense blood flow and respond to it in very specific ways.
Okay, I was imagining in the different part of for example, brain cell or some hand cell, different kind of cell have a different ambient flows. And since you are saying that the cell wall dependent on the flows, and cell wall decide the shape and size, so that's why we see different kind of a structure of the cells in the body, or maybe some different thing. I was thinking that.
Well, I don't know, but perhaps we'll have to ask people to look at look at that kind of a correlation. Yeah.
Thank you.
Yeah, so maybe, yeah.
Yeah.
So I think maybe I missed something. So like you talked of this like different increasingly complex model for the cell membrane, right?
Right.
So the present understanding is somehow that the structure is maintained by coupling and sort of active elements in the actin filament.
Right.
So functionally, I mean, what does this, I mean, like why is this, I mean, does it have any significance? In terms of what the functions of the, like these rafts and the cell membrane.
Right. So I think I tried to mention that at the end. The composite membrane, I mean, the actin composite membrane can be exquisitely regulated, right? Because there's a machinery that's consuming ATP and energy to drive the composition in a particular way. And that is now tuning the response of the cell to the external environment. So if you now, let's say, get rid of this machinery, the response of the cells to the external environment is, dramatically different. And and this is manifest in many different conditions that we have looked at, and I'd be happy to chat with you about that. Yeah.
Okay, Spender.
Hi, great talk. Just maybe you said it, but I missed it. But if you look at the cell in the brain, the nerve cell, then we know that it performs very complex tasks, actually. So is the cell membrane there also very similar, or is there some big differences?
Yeah. So, well, the neuron is, or neurons, I mean, actually, first of all, there are many cell types in the brain, and there are many types of neurons. And the architecture of neurons is determined by the actin cortex, the architecture, the physical shape and form. The membrane itself, because it has the same elements of actin and all the machinery that do make this composite, the membrane is also going to exhibit the same kind of membrane composites. Today, we don't understand the implications of that because that system is, in my view, very complex to address. Because, first of all, it has this exquisite and extraordinary architecture. And then, what does it make sense to look at a little piece of it and see what it does, or should one be looking at the whole thing? So I think, for myself, I think I have to pose the question in a different way and see whether these machineries operate in very specific contexts. And this is something that we are going to do. But I think, every cell type will have a similar question. And I think the point that was being alluded to earlier, are these machineries functioning in different cell types and what cell types work? The point I want to make is that the machinery will operate wherever cells have these components. And the cells will use them because they consume energy and they are generating, I would say, information modulation scaffolds. So in the neuron, which is the ultimate information transmitting and receiving system, I'm sure it's going to use these things.
Thank you.
Mahesh.
Thanks, Jitu, for the very nice talk. I don't have a question. I just wanted to add a minor historical footnote, since specifically you use the term poster person. At the very beginning of your talk, you reference Agnes Pockels. So she's the first person in history to have measured surface tension.
Right.
And she did it, women were not allowed in the university at the time. Her brother, who was a university student, used to smuggle books. And she sent a letter. She sent a letter to Lord Raleigh, who had it published as is in Nature.
Right.
And the paper that preceded her work was by James Clerk Maxwell, and the following paper, the one that followed her, was by Van der Waals. And the only correction I would offer is Langmuir trough is not by Langmuir, it's by Pockels. It was called the Pockels scale, which was improved by him together with Catherine Blodgett.
That's right, yeah. No, the Pockels cell was the forerunner of what Langmuir used, which came to be called the Langmuir trough. But anyway, you're right. I mean, thanks for that correction, but I didn't displace the history. She was the forerunner of all these
Actually,
measurements, yeah.
Lord Rayleigh continued her experiments in 1891. He did a very beautiful calculation. So he introduced a drop of oleic acid. He knew the amount of oleic acid he introduced on the surface of water. He made an assumption that it was a monomolecular layer, using which he back calculated or estimated what would be the size of
The
an
lipid.
atom or a molecule for the
Right.
first time as 10 angstroms.
Right. Right. In fact, that number was used by those Gödel and Gretton to look at the bilayer.
Okay, good. Thanks for all the questions, and let's thank Jitu again for a wonderful talk.